Dear student, I am Dr. Prakash Mungli, Professor of Biochemistry. In this lecture, I will be explaining you all about glycoproteins. Let's get into the details. Half of our body proteins are glycoproteins, and the attachment of glucose or a carbohydrate moiety onto a proteins can be enzyme mediated, wherein enzymes are catalyzing the addition of carbohydrate over a protein's surface, or attachment of carbohydrate over a protein can be non-enzyme mediated. So, the glycosylation process is enzyme mediated, whereas the glycation process is a non-enzyme mediated process. Note the difference in the wording here. So, that is the one that will help you in understanding. Whenever you look at the terminology, you will understand whether it is an enzyme mediated process or non-enzyme mediated process. So, if you see a term glycosylation, you know that it is an enzyme mediated process. Enzymes will attach carbohydrate over a protein, that referred as glycosylation. Whenever you look at the term glycation, that is a non-enzyme mediated process. That's how the terminologies will help in understanding how a carbohydrate is added over the surface. We're going to look at examples later. So, all human plasma proteins are glycoproteins except albumin, that is present in the plasma. In fact, albumin is a major plasma protein, but it is not a glycosylated protein. Now, the carbohydrate content present in the glycoprotein, it can vary from 1% to 85% weight. Compare this with proteoglycans. Proteoglycans, they contain high amount of carbohydrate, almost about 95% of their weight will be carbohydrate. Unlike the glycoproteins here, the carbohydrate content can be 1% or it can go all the way up to 85%. Now, in the glycosylation process, the specific glycosyl transferases will transfer carbohydrate moiety over the surface of proteins. The glycoproteins, they have a diverse function and bring change in the cellular activity. That's why addition of carbohydrate over a protein molecule, it is referred as a sugar code of life. And also the study about carbohydrates present over proteins is referred as glycomics. And glycomics helps us to study what are the diverse functions of proteins which we have not yet explored. Now let's look at some of the functions of glycoproteins present in the plasma and also present in the extracellular matrix. Collagen is a glycoprotein present in the extracellular matrix and it is a structural glycoprotein. Mucins are also glycoproteins which are acting as a lubricants and protective agents. Transferrins and ceruloplasmins. Transferrin is a iron transferring protein that's present in the plasma. Ceruloplasmin is a copper storage and copper transfer protein. Similarly, like we have immunoglobulins which will protect against invaders, provide humoral immunity to us. Histocompatibility antigens, these are the proteins that are participating in immunological mechanism. Then we have certain hormones like chorionic gonadotropin, thyroid stimulating hormone, and variety of enzymes that are present in our cells inside or present outside the cell. They are all glycoproteins. And various proteins that are involved in cell-cell communication, cell and virus, cell bacterium, cell and hormonal interaction. All the receptors, transmembrane proteins almost all of them are glycoproteins. Like that, we have lectins, calnexin, calreticulin, cell surface proteins, and so many others. So there are a lot of glycoproteins in our body and they will bring diverse functions. Now let's look at functions of oligosaccharide chains that are present over glycoproteins. So I have explained most of it in the previous table. Let's go over some more additional information in this particular slide. So they bring change in physiochemical properties of the protein. Having a carbohydrate moiety present over a protein molecule, it brings in a solubility, viscosity, charge, conformation, and denaturation process. So this type of change in physiochemical properties can brought in by the presence of carbohydrates. And also having carbohydrates over the surface of proteins provides binding sites for various molecules as well as like bacteria, viruses and parasites. And these glycoproteins, the carbohydrate moiety over glycoproteins present over cell surfaces, they will help in recognition of signals. They protect against proteolysis, and also carbohydrates, they ensure the correct folding of proteins that are exported from the cell and target incorrectly folded proteins for the catabolism. And also carbohydrates present over protein protect peptide hormones and other plasma proteins against clearance by the liver. They permit anchoring of extracellular proteins in the cell membrane and of intracellular proteins inside subcellular organelles like endoplasmic reticulum and Golgi complex. And also carbohydrates being present over the proteins, they direct intracellular migration, sorting and secretion of proteins, and they affect embryonic development and cell and tissue differentiation. They may also affect sites of metastasis selected by cancer cells. So there are variety of functions that is given out by having a presence of carbohydrate over our protein molecule. That's the importance of having oligosaccharide chains over glycoproteins. Coming to structure of glycoproteins. The carbohydrate moiety present over glycoprotein, it is short and branched heteropolymer. Remember that. Compare this with the proteoglycans containing 𝐒𝐵𝐼𝐶. You know, 𝐒𝐵𝐼𝐶 is a repeating disaccharide and it is an unbranched polymer of carbohydrate. So here glycoprotein's carbohydrate chain is very short and it is highly branched. We are going to see how. Now there are 8 commonly found monosaccharides present over glycoproteins, and those are glucose, galactose, mannose, fucose, xylose, N-acetylglucosamine, N-acetylgalactosamine and N-acetyl-neuramic acid, which is referred as sialic acid. And also note that N-acetyl-neuramic acid is abbreviated as NA. And some of these carbohydrate moiety means some of these monosaccharides are further sulfated, like galactose, N-acetylgalactosamine and N-acetylglucosamine can be further sulfated. So having this acetyl group attached to a monosaccharide and also having further being sulfated, it will bring a negative charge to a glycoprotein molecule and make them a polyanions. The process of glycosylation, it begins in the lumen of rough endoplasmic reticulum and it will continue in the Golgi apparatus. And the nucleotide sugars are the donors of monosaccharides that are present over glycoproteins. Also note that whenever a carbohydrate is added over a protein molecule, whether it is a glycoprotein synthesis or proteoglycan formation, wherever the carbohydrate is being added, or wherever carbohydrate is transferred, it has to be there in a nucleotide sugar form. A nucleotide it can be uridine diphosphate UDP, or it can be guanosine diphosphate GDP, or it can be CMP cytidine monophosphate. So this type of nucleotides are the ones which will bring monosaccharides into the reaction, and thereby only the carbohydrate is added and the nucleotide will be released. And this is what is necessary for a glycosylation process. Remember that. Nucleotide sugar is a general term. Now let's move on to see what are the different types of nucleotides that are bringing different types of monosaccharides into the reaction. So let's look at UDPs, uridine diphosphate. Uridine diphosphate is bringing glucose as UDP glucose, UDP galactose, UDP N-acetyl glucosamine, UDP N-acetyl galactosamine, and also UDP xylose. That means it brings 5 types of monosaccharides into the reaction. And whereas mannose and fucose, it will be brought in by GDP, guanosine diphosphate, and the sialic acid, it is brought in by CMP, that is sialic acid is, as I said earlier, it is N-acetyl neuraminic acid. So CMP NANA means cytidine monophosphate N-acetyl neuraminic acid. So these are the types of nucleotide sugars that are coming into the reaction to make glycoproteins. Now this table is showing you how the sugar units are brought in to the reaction, the type of exos, abbreviation, sugar nucleotides, and also some of the comments related to that specific type of sugar unit that is brought in. Galactose is an exose, it is brought in as UDP galactose, glucose as UDP glucose, and then N-acetyl galactosamine brought in as UDP N-acetyl galactosamine, N-acetyl glucosamine brought in as UDP N-acetyl glucosamine, and then xylose brought in as UDP xylose. And note that this UDP xylose, it is making a linked trisaccharide in a O-linked glycoproteins. We are going to see that later. And then the GDP brings mannose and fucose, and the CMP brings N-acetyl neuraminic acid or the sialic acid. So these are some of the things that you need to remember in terms of nucleotide sugars that are coming into the reaction. Now let's look at major classes of glycoproteins. Now the glycoproteins can be classified depending on how they interact with the protein. So there can be 2 types of interactions based on the linkages. One is a O-glycosidic linkase containing glycoproteins. Other is N-glycosidic linkase containing glycoproteins. As it is shown here in the figure, O-linked glycoproteins, they are going to interact with amino acid serine which contain hydroxyl group. So the carbohydrates will be added over the hydroxyl group of a serine which is part of a polypeptide chain. That's how O linkages are done. Of course, there are certain exceptions where O linkages with the type of carbohydrate that is added to the serine can vary from a type of different types of the glycoproteins. And now in the N linkase, the amino acid asparagine with its side chain amino group is the one that will accept carbohydrate to be added over a polypeptide chain. That's how N linkages are done. So this is how major classes of glycoproteins can be done. One is O-linked glycoproteins and other is N-linked glycoproteins. Now the other class of glycoprotein is a glycoprotein that uses glycophosphatidyl inositol as an anchored molecule. So that means the type of glycoproteins that we classify here is GPI. GPI means glycophosphatidyl inositol, which is attached to the cell membrane, and further the GPI is going to anchor a protein. So GPI bound proteins are referred as GPI anchored glycoproteins. So let me just go over a little more into the composition of the GPI here. It is important for you to understand how a GPI is built and how it is useful in anchoring a protein over the cell membrane. So as you see here, this entire structure that you are seeing, starting from the ethanolamine, mannose, glucosamine, inositol and fatty acid that is attached. So start from the additional fatty acid there, which is attached to the plasma membrane, and then it is attached with inositol. Go from there, glucosamine, inositol is attached with the glucosamine, and then 3 mannose molecules, and one of the mannose is attached with ethanolamine, and then last mannose with the phosphate bond attached with the ethanolamine, and that ethanolamine is going to interact with a protein via glycine. So that's how a glycoprotein, which is attached over the surface of a membrane using GPI anchor. So this is the GPI anchor that you are seeing, that I am writing here. This entire structure is a GPI anchor, remember. So the GPI anchor is referred as glycophosphatidyl inositol. As you see, glycophosphatidyl, glyco means it has carbohydrate like mannose, glucosamine. This is our carbohydrate. Phosphatidyl, it is phosphorylated, inositol phosphate. See, inositol, P is there, phosphate is there. Glycophosphatidyl inositol is the one that is attached to the membrane with the fatty acid, and then carbohydrate moiety, and then ethanolamine, and that is going to attach with the protein. That's how a protein is attached to the cell membrane. And the GPI anchor, since it's little longer in size, so it will give a freedom of movement to a protein that uses GPI anchors to attach to the plasma membrane. And that is how some of the proteins will be attached to the plasma membrane, and those are referred as GPI anchored glycoproteins. We are going to go into that later, and also we are going to study a disease related to this. If there is any mutation in a gene coding for any of the component here in this GPI composition, that is, it can be ethanolamine, mannose, glucosamine and inositol. So all these molecules are brought in by specific enzymes to build that GPI over the cell membrane. If there is any mutation in enzymes that are bringing all these molecules to build GPI, if that is defective, if building of GPI is defective, then all the proteins that uses GPI anchors to be present over cell membrane won't be able to be added over the cell membrane, and that may lead to issues and how the cell functions, how our body functions overall, right? And that is studied with using a disorder called paroxysmal nocturnal hemoglobinuria. We are going to study this disease to understand how important this GPI can be, because if there is an issue in synthesis of this GPI, it will lead to paroxysmal nocturnal hemoglobinuria. We'll go into that later. Now let's look at synthesis of O-linked glycoproteins. The polypeptide chain of the glycoprotein is synthesized first, and then later on the individual sugar units are sequentially attached to the polypeptide chain one at a time. That's how O-linked glycoproteins are made. That means for synthesis of O-linked glycoproteins, first polypeptide chain has to be made, which is synthesized over ribosomes present over rough endoplasmic reticulum, and then it is entering into the lumen of rough endoplasmic reticulum, and then the further modification will be going on where each individual sugar units can be added one at a time. That's how O-linked glycoproteins are made. Whether it is in the lumen of endoplasmic reticulum or in the free ribosomes in the cytoplasm, so protein has to be synthesized first. So, synthesis of the polypeptide chain. Let's look into that first, and then we will see attachment of sugar units over that polypeptide chain. So, polypeptide chain is synthesized or ribosomes present over rough endoplasmic reticulum, and then it enters into the lumen of rough endoplasmic reticulum. So, in the lumen of rough endoplasmic reticulum, and also as it moves on to the Golgi complex, the oligosaccharide chain is built by sequential addition of carbohydrates one at a time, which are coming into this reaction in the form of sugar nucleotides that we have seen in the previous slide, depending on which monosaccharide is coming in. Example, if glucose is brought in, it will come in as UDP glucose, right? And this type of sugar units are brought in by specific glycosyl transperases. So, this is how a glycoprotein is built. First protein is made and then sugar units are added one at a time. Okay. So, overall synthesis of O-linked glycoprotein as it is written in the table on the right hand side. So, it involves a battery of membrane bound glycoproteins, glycosyl transperases acting in a stepwise manner. Each transfers generally a specific and a particular type of monosaccharides to link in a particular linkage. And the enzyme involved in this process are present in the subcompartments of Golgi apparatus. That's where most of the glycolysis is going on. And each glycosylation reaction involves an appropriate nucleotide sugar which is coming in. And note that the role of dolichol pyrophosphate is not there here in O-linked glycoprotein synthesis, unlike N-linked glycoprotein synthesis that we are going to see later. And also there is no role of glycosidases, like removal of a carbohydrate which is already attached. And also glycoprotein synthesis is not inhibited by 𝗠𝐞𝐝𝐢𝐴. This is something that differentiates O-linked glycoproteins from N-linked glycoprotein synthesis, because in N-linked glycoprotein, we are going to see later, there is a role of dolichol PP, that there is a role of glycosidases, and also there is a role of 𝗠𝐞𝐝𝐢𝐴, where N-linked glycoprotein synthesis is inhibited by 𝗠𝐞𝐝𝐢𝐴. Such things are not associated with O-linked glycoproteins. And also O-glycosylation occurs post-translationally, especially at serine and threonine residues, because serine and threonine, these are the hydroxyl containing amino acids. Now let's move on to see N-linked glycoproteins formation. N-linked glycoproteins compared to O-linked glycoproteins, these are the major class of glycoproteins. That's the first point that you should remember here. Majority of the glycoproteins that are present in our body, they are N-linked glycoproteins. So here a polypeptide chain is synthesized on the ribosomes present over rough endoplasmic reticulum, and the mannose-rich core of oligosaccharide is constructed over dolichol pyrophosphate, which is attached to the rough endoplasmic reticular membrane towards the cavity or towards the lumen. So that means a polypeptide chain and the oligosaccharide chain, both of them are synthesized simultaneously. As the polypeptide chain is synthesized over ribosomes present over rough endoplasmic reticulum, in the lumen of rough endoplasmic reticulum, oligosaccharide chain is built. Okay, so as the polypeptide chain is entering into the lumen of rough endoplasmic reticulum, mediated by that signal sequence, so on the other side, in the lumen, especially attached to the luminal side of the rough endoplasmic reticulum, there is a molecule called dolichol, which is phosphorylated, called dolichol pyrophosphate. So it is dolichol PP. PP means pyrophosphate, two phosphates attached to dolichol, and it is attached to the membrane of rough endoplasmic reticulum and towards the luminal side. And note that this dolichol pyrophosphate, in fact, it is a derivative of a cholesterol metabolism intermediate called farnesyl pyrophosphate, and this farnesyl pyrophosphate, it is coming from mevalonate in the cholesterol biosynthesis. So farnesyl pyrophosphate, as it is synthesized in the cholesterol biosynthesis, it can be diverted into cholesterol, but same farnesyl pyrophosphate can also be diverted into dolichol formation. So that dolichol is the one which is involved in oligosaccharide chain synthesis, which is required for N-linked glycoproteins synthesis. Okay, so the core oligosaccharide chain, once it is built, it will be transferred from the dolichol pyrophosphate to a newly synthesized polypeptide chain, especially over amide nitrogen of asparagine residue. Remember, the oligosaccharide chain is attached to the side chain nitrogen, that is the amide nitrogen present in an asparagine amino acid in a polypeptide chain of a protein molecule. That's the difference between synthesis of O-linked glycoproteins over N-linked glycoproteins. As you know from the previous slide, in O-linked glycoproteins, once the polypeptide chain is synthesized and in the lumen of endoplasmic reticulum and more predominantly in the Golgi apparatus, one sugar unit is attached at a time sequentially to make or to build that carbohydrate chain in O-linkage, and these sugar units are coming in as nucleotide sugars. Whereas in N-linked glycoproteins, so polypeptide chain is synthesized over rough endoplasmic reticulum and coming into the lumen, and as the polypeptide chain came in, on the other side an entire oligosaccharide chain is built on dolichol pyrophosphate, and that entire oligosaccharide chain, once it is built, it will be transferred to asparagine residue of a polypeptide chain, especially the amide nitrogen or the side chain amino group of asparagine is going to accept that oligosaccharide chain which is built somewhere else, that is on dolichol pyrophosphate. Okay, so that is the difference here between synthesis of O-linked glycoproteins and N-linked glycoproteins. So here is how the role of dolichol pyrophosphate is shown in N-linked glycoprotein synthesis. So here is the dolichol, which is phosphorylated dolichol pyrophosphate, over which UDP N-acetyl glucosamine is added first, and then all the nucleotide sugars coming in over this is going on over dolichol. It is not yet transferred to a protein molecule. Over dolichol, a sugar unit is built. So very first sugar unit that is added over dolichol, which is attached to the rough endoplasmic reticular membrane, is N-acetyl glucosamine, which is coming in as UDP N-acetyl glucosamine. This will be inhibited by a 𝗠𝐞𝐝𝐢𝐻𝐼𝐶. 𝗠𝐞𝐝��𝐻𝐼𝐶 inhibits the synthesis of dolichol pyrophosphate oligosaccharide, which is necessary for making N-linked glycoproteins. So that's the another difference that we see with compared to O-linked glycoprotein synthesis. So one molecule of N-acetyl glucosamine added over dolichol PP, and then sequentially another molecule of N-acetyl glucosamine is added, and then the mannose will be added. So one more mannose. So few more mannose added. So now we have 5 mannose attached, and then further mannose being brought in, and then this is how a oligosaccharide chain is built sequentially. And once the sufficient oligosaccharide chain is built, as you see here, 2 molecule of N-acetyl glucosamine, and then so now once a sufficient oligosaccharide chain is built over dolichol pyrophosphate, and this oligosaccharide chain is transferred en masse, it is transferred to asparagine amino acid over a polypeptide chain. So it will be added on to the amide nitrogen present in the side chain of asparagine. So this is what is the oligosaccharide chain that is built. So it has 2 N-acetyl glucosamine, 9 mannose molecules and 3 glucose molecules, and that oligosaccharide chain is transferred. That's how a carbohydrate moiety is added over asparagine residue in N-linked glycoprotein synthesis, and also you should remember the role of dolichol pyrophosphate in building the oligosaccharide to make N-linked glycoproteins. Okay, so here it is shown protein getting into the lumen of endoplasmic reticulum, and the oligosaccharide chain is built on dolichol pyrophosphate. As it is shown, there is a dolichol pyrophosphate here. Oligosaccharide chain is built, and then that entire oligosaccharide chain is transferred to a protein molecule, especially the asparagine amino acid having the amide nitrogen there in the side chain amino group, and then that oligosaccharide chain further it will undergo modification as it is moving on from rough endoplasmic reticulum to the Golgi complex, and before it is being secreted, the carbohydrate chain can be modified. That's where glycosidases will come and they will remove some of the monosaccharides, and modification or further addition of different types of monosaccharides can go on. So, processing and trimming process will be going on in the rough endoplasmic reticulum. So, as it is shown in the figure here. Now, so in the synthesis of N-linked glycoproteins, so following steps are involved. First is synthesis of oligosaccharide unit, which is happening over dolichol pyrophosphate as I have explained earlier. And then the synthesis of polypeptide chain is going on simultaneously, and once these two things are ready, then the transfer of oligosaccharides from dolichol pyrophosphate will go on, and that is done by specific protein oligosaccharide transferase enzyme. And then trimming and addition of monosaccharides will be going on in the Golgi apparatus. And note that 𝐒𝐲-NEXin, which is a chaperone protein, and ERP57, that is homologous of protein disulfide isomerase, will ensure proper folding of glycoproteins, which are N-linked type here. Now the types of N-linked glycoproteins based on the complexity of monosaccharide units that are attached over the glycoproteins, especially the N-linked glycoproteins, can be classified into three major classes. So the N-linked glycoproteins can be of complex, high mannose or hybrid. As you see in complex, there are a variety of monosaccharide units that are present with the figure color coding you can see. So in complex N-linked glycoproteins, you will see sialic acid, galactose, and then mannose, N-acetyl glucosamine. So all kinds of monosaccharide units can be seen there. And in the hybrid, it's a mixture of monosaccharide units. So in high mannose, most likely you will see higher number of mannose residues. Now all three types of N-linked glycoproteins, they contain common pentasaccharide, that is, they contain 3 mannose and 2 N-acetyl glucosamine bound to asparagine, but they differ in outer chain as you see here. So this common pentasaccharide is there for all three. That means there are 2 N-acetyl glucosamine and 3 mannose residues, which is common for all. Now N-acetyl lactosamine units are often found in N-linked glycan chains. Coming to main features of glycoproteins. So oligosaccharide chain that is transferred to a protein molecule, especially over asparagine residue, it is composed of 2 N-acetyl glucosamine, 9 mannose residues and 3 glucose residues. That is the sufficient oligosaccharide chain which is built over dolichol pyrophosphate, and then that oligosaccharide chain is transferred to asparagine residue. And note that the synthesis of this type of oligosaccharide chain, it is inhibited by tunicamycin. Now a transfer of oligosaccharide chain occurs over asparagine residues in the sequence as asparagine X serine 309, where X is any residue except proline aspartate or glutamate. This is the sequence of amino acids where as oligosaccharide chain is attached to an asparagine residue in a polypeptide chain. It will be asparagine, any other amino acid, serine or threonine, that type of sequence in a polypeptide chain. And the transfer can occur co-translationally in the endoplasmic reticulum as the protein is coming out of a ribosome, entering into the lumen of endoplasmic reticulum, and in the lumen of endoplasmic reticulum this oligosaccharide chain can be transferred because it is already built over dolichol pyrophosphate. Now the protein bound oligosaccharide is then partially processed by glucosidases and mannosidases. If no additional sugars are added, that will result in high mannose chain, and if processing occurs down to the core pentasaccharide, that is 3 mannose and 2 N-acetyl glucosamine, the complex chains are synthesized by addition of N-acetyl glucosamines, few more, or removal of 2 mannose and the stepwise addition of additional N-acetyl glucosamine. So like this, the modification of that oligosaccharide chain can go on to make different types of N-linked glycoproteins. Now once all this process goes on, so the cellular targeting of these glycoproteins need to be done. Whether it is a O-linked glycoproteins or N-linked glycoproteins, they need to be targeted to specific subcellular organelles or to the cell membrane, and that will be done by chemical markers that will help in targeting of glycoproteins to wherever they need to go, whether they need to go to lysosome or to mitochondria or to the nucleus or to the cell membrane or even to get out of the cell. So there must be a chemical markers for targeting process. So the cellular targeting, the chemical markers that are needed for that process, it has to be done in the either in the endoplasmic reticulum or in the Golgi complex, so that these proteins are targeted. So in order to understand this concept of cellular targeting of glycoproteins, let's take example of how proteins that are meant to go to the lysosome, proteins that are targeted to the lysosome, like acid hydrolases that are present in the lysosome, how they are sent from the rough endoplasmic reticulum through the Golgi complex, how they will go to lysosome. So let's understand that concept. So targeting of glycoproteins like acid hydrolases to the lysosome. So the chemical marker for proteins that are meant to go to lysosome is mannose 6-phosphate. Remember that. And how this mannose 6-phosphate will be added over a protein that is being targeted to lysosome. It will be done by, so this process will be initiated by an enzyme called glucnag phosphotransferase. This glucnag phosphotransferase is N-acetyl glucosamine phosphotransferase. Let's see what this enzyme does. Now consider a glycoprotein being synthesized on ribosomes over rough endoplasmic reticulum, entering into the lumen of rough endoplasmic reticulum, going into Golgi complex, and if this glycoprotein has to go to lysosome, so this protein having a mannose residue, especially it's an N-linked glycoprotein. As you know, majority of our proteins in the body are N-linked glycoproteins, so it will have a mannose residue over it. So this mannose residue, it will be further added with N-acetyl glucosamine, which is coming into the reaction as UDP N-acetyl glucosamine. So over a mannose, N-acetyl glucosamine is added. As you see here, I am marking it. So we have protein which is attached with mannose, and that mannose is further attached with N-acetyl glucosamine. This type of attachment is initiated by N-acetyl glucosamine phosphotransferase. Remember the name here. This is the most important enzyme that you need to remember, because deficiency or defect in this enzyme because of mutation in the gene that is coding for N-acetyl glucosamine phosphotransferase, it will give rise to a disease called eye cell disease. That's why this enzyme is important, you should remember. So once we get a protein which has got mannose and further added by N-acetyl glucosamine, but note that the chemical marker for proteins that are destined to go to lysosome is only mannose 6-phosphate, not mannose 6-phosphate N-acetyl glucosamine, which is being added there right now. You are seeing there, this one. That N-acetyl glucosamine has to be taken out, only the mannose has to be phosphorylated at 6th carbon there. So that is done by removal of N-acetyl glucosamine from the mannose 6-phosphate. That the enzyme that removes N-acetyl glucosamine is phosphodiesterase, which is breaking that phosphodiester bond. So this part is taken out. Only N-acetyl glucosamine will be released, and now your protein is phosphorylated at 6th carbon of mannose. So that is the chemical marker which is added on a protein. So this protein is now ready to be taken to lysosomes. So this is how a chemical marker is added over a protein molecule, so that the protein is targeted to a different subcellular location. So the chemical marker for lysosomal proteins is mannose 6-phosphate, and this is how mannose 6-phosphate is added. And this mannose 6-phosphate present over protein which is targeted to lysosome will bind to mannose 6-phosphate receptors present on trans-Golgi, and that trans-Golgi is now will become secretory vesicles, and that will be targeted to lysosome. So and then it will be taken to lysosome, where it will be internalized, where the protein is internalized, and that's how a protein reaches lysosome with the help of mannose 6-phosphate interacting with the mannose 6-phosphate receptor. And that is what is shown here in the figure. A protein, N-linked glycoprotein, is synthesized on the ribosomes, entering into the rough endoplasmic reticulum, glycosylated. So pre-lysosomal enzyme there, and then N-linked oligosaccharide chain is built, and then a chemical marker is added. Especially it happens either in the endoplasmic reticulum or in the cis-Golgi. And the most important enzyme which initiates the addition of chemical marker is N-acetyl glucosamine phosphotransferase. Once the chemical marker is added, where a phosphorylated mannose, especially the 6th carbon, is phosphorylated, which is a chemical marker for an enzyme to go to lysosome. So it is going to go and bind to mannose 6-phosphate receptor present over trans-Golgi, and that is now directed in the form of transport vesicles. It will go and fuse with the lysosome, where over the lysosomal membrane it's going to recognize that receptor which is bound with mannose 6-phosphate containing protein, which is an enzyme now, and only the enzyme is internalized, a receptor is recycled, and that's how the enzyme has reached lysosome with the help of mannose 6-phosphate. Okay, deficiency in this process, whether a deficiency in attachment of mannose 6-phosphate over a glycoprotein which is targeted to lysosome, if there is a defect there, especially if a glucnac phosphotransferase enzyme is mutated, it might lead to eye cell disease. Or if there is a mannose 6-phosphate receptor itself, if it is defective, it can also lead to similar condition like eye cell disease. So eye cell disease is where the enzymes that need to be present in the lysosome for degradation of all kinds of material are failing to reach there because mannose 6-phosphate is not added, right? And what happens to the lysosomes at that time when lysosomes don't have enzymes to degrade all kinds of materials dumped into the lysosome? So it won't be degrading all those material like carbohydrates, proteins, lipids. They are not degraded because lysosomes don't have these acid hydrolases. Why? Because they are not coming in there. Why? Because mannose is not phosphorylated at its 6th position to make mannose 6-phosphate, which is a chemical marker for all the proteins to go to lysosome which are meant to go there, right? So this type of issues can happen leading to disorders. So that's all about details on glycoproteins. I hope this lecture has helped you in understanding two main types of glycoproteins, that is O-linked glycoproteins and N-linked glycoproteins, and all the details related to that. Thank you.